Welcome back to the International Virtual Conference 2020. I would like to call Dr. Lakshmi Ma'am, Assistant Professor, Department of Chemistry, to introduce the guest speaker of the session 6, Dr. Ananda Kumar Pandi sir. Over to Dr. Lakshmi Ma'am.
Thank you, sir. Good morning to one and all present here. I am Lakshmi, Assistant Professor, Department of Chemistry. With great pleasure, on behalf of management, principal, vice principal, HOD, staff members, and students of Anna Violet Arts and Science College, introduce our guest speaker of today's session, Dr. Ananda Kumar Pandi, Assistant Professor, Department of Biochemistry, All India Institute of Medical Science, Diohar, Jharkhand, India. Our guest speaker today is one of the finest diamonds of University of Madras. He has acquired his master's degree in the year 2004 and PhD in the year 2009 in medicinal biochemistry from University of Madras. He has more than 12 years of teaching experience and 10 years of research experience. He is one of the editorial board members of American Journal of Medicinal and Biological Research, Science and Education published in USA. Jacobs Journal of Enzyme Engineering USA, Journal of Natural Products and Biomedical Research India, Journal of Pharmaceutical and Biological Sciences India, and Associate Editor in International Journal of Health and Pharmaceutical Research. He is also an advisory board member for Advanced Journal of Pharmacy and Life Science Research India. In addition, he reviewed articles for nearly 8 international journals, added to which he has more than 25 international and 15 national publications. He is a member of various professional bodies like Asian Pacific Chemical, Biological and Environmental Engineering Society, Hong Kong, Asian Council of Science Editors, Dubai, UAE, Indian Association of Biomedical Sciences, India, Society of Biological Chemists, Indian Institute of Science, Bangalore, India. We welcome you, sir, for the session to deliver your lecture on carcinogens, in which we all would be highly interested. Kindly deliver your lecture, sir, please.
Okay. Thank you, Dr. Am I audible? Hello?
Yes, sir, you can go ahead, sir.
Okay. Thank you, Dr. Lakshmi, Department of Chemistry, for your for a nice introduction.
Thank you, sir.
So, good morning, one and all. Present for the for this international virtual conference on recent advances in biochemical and material sciences. On the outset, I would like to congratulate and appreciate the management, principal Dr. Kamala Balakrishnan, and the convener of this international webinar, Dr. Parimala, co-conveners Dr. Prabhakaran, Dr. Chandramohan, and the entire organizing team for bringing this international virtual conference. This is really a wonderful platform for the students, the faculty, and the other interested scholars to interact with the different researchers all over the world, and the theme of the topic is also very well relevant in today's life. It's recent advances in biochemical and material sciences. So with this I would like to start the session on the topic. So today's topic is I'm going to talk on carcinogens, the key role players in cancer. See, although we are in this COVID-19 pandemic and the extensive research has been carried out on this COVID-19, although we are in this pandemic, I think this topic, carcinogens, the key role players in cancer, is a very important topic to discuss, because as we all know that cancer is a very deadly disease, and we know the mortality rate is quite high in cancer. And if you see the scenario, the world scenario, the statistics itself, more than 9.7 million cases are detected every year, and about 20.4 million people are living with cancer in the world today. It's really although it's a bad news, no, to begin with, but we have to accept the fact that the statistics says that 20.4 million people are nearly affected all over the world today because of cancer, and 6.7 million people, they are predicting that more than 6 million people are going to be affected. And by 2025, the statistics says that 15 million people will be, the mortality rate may increase more than this. Okay. And talking about the most common cancer in the world. So this, so the most common cancer, if you see, it is the lung cancer, which is the most important cancer. The first, which comes the first, lung cancer is the most important common cancer in the world. Then comes the breast cancer and the colorectal cancer. And if you come to the Indian scenario, in the Indian scenario, more than 15 lakh people are affected every year, and more than 5 lakh people are dying in India every year because of this deadly disease. And we know that in India, the most common cancer is breast cancer, and followed by that, it is the cervical cancer and oral cancer. So with this brief introduction on cancer, we'll move on to, so talking about the age. So is there do this cancer patients are really, it's not like diabetic or other diseases. Cancer, it's right from the newborn until the death, people can be affected because of cancer. But talking about the age, yes, newborn baby is affected because of cancer. Yes, there are babies who are born with cancer. So for example, some cancers like neuroblastoma, or about, they are saying about 5% of the babies are, you know, born with some cancer like neuroblastoma detected in that stage, early stage. So it's such a deadly disease. So just before going to that, we'll have an introduction about what is cancer and what are the causative agents. Then we'll move on to the topic exactly about carcinogens. So we all know that the term cancer applies to group of diseases in which cells grow abnormally. So the term cancer is a broad term where we apply this for denoting which says that for the abnormal cell proliferation. So the cancer is a common term. So if you see the term tumor represents a mass of tissue which is formed as a result of abnormal, excessive, uncoordinated, autonomous, and purposeless proliferation of cells. So although cancer is a very broad term, tumor refers to the proliferation of cells of that mass of that tissue. And the science dealing with the study of this tumor or neoplasm is called as oncology. So if we go to, if we see in multi-specialty hospitals, there is a separate department for oncology, and the physician who is treating this, who is involved in the oncology department, they are called as oncologists. So the word oncos come from tumor, and logos means study. So differentiating this benign versus malignant tumors. So tumors can be of two types. One is the benign tumor, the other one is the malignant tumor. So benign tumor, the major difference between the benign and malignant is benign tumors, they does not invade or spread to distant sites in the body, and it does not destroy the tissue in which it originates. So examples, so benign tumors are generally considered as non-cancerous tumors. We don't use the term cancer here, although we say benign tumor. So they are classified under non-cancerous tumor. For example, moles, uterine fibroids. So these are all classified under benign tumors. Coming to malignant tumors. So the malignant tumors, it invades and destroys the tissue in which it originates. So it can spread from a primary site to a different site in the body via the bloodstream. So the major difference between benign and malignant is that malignant tumors have the ability to spread from one site to other via the bloodstream and the lymphatic system. So this term is usually referred to as metastasis. So we use the term metastasis for this, where the tumor spread from one part of the cell to other. So the term used for all malignant tumors. So this malignant tumors is we commonly called as cancer. The term used for all malignant tumors is cancer. Example, we all know liver cancer, lung cancer, breast cancer, colon cancer. There are different types of cancer. So there's a major difference between, even if you see the diagram, you can easily understand the major difference between the benign and malignant tumors. Malignant tumors, they invade the neighboring tissues, they enter bloodstream, and they metastasize to different stages. That is very important. So they have the capacity to invade the neighboring tissues by entering the bloodstream or the lymphatic vessels, and they metastasize to different sites. So this is the difference between benign and malignant tumors. Talking about the difference between the normal and the cancerous cell. So what are the characteristics of the major characteristics between the normal cell and the cancerous cell? So if you see the normal cell, normal cells are usually small, uniform shaped nuclei. Relatively, they have a large cytoplasmic volume. If you see the diagram here. So if you see the cancer cells, on the contrary, they are very large. They're variable shaped. They have variable shaped nuclei, and their cytoplasmic volume is smaller. And similarly, if you see the size of the cell size and shape, so here it is, the cells are arranged properly. We have a proper arrangement of cells in case of normal cells, but here we see a disorganized and distorted cell arrangement in case of cancer cells. Apart from this, normal cells, they have a differentiated cell structure, and they properly present their cell surface markers for the intracellular communication or inter and intracellular communication. But if you see here on cancer cells, there is loss of all these loss of the normal specialized feature, as well as elevated expression of certain cell markers. So this, apart from that, if you see the normal cells, they are lower levels of dividing cells. We cannot say lower level. The division is regulated. There is a regulation in the division of cells in case of normal cells, and the cell tissues are clearly demarcated. But whereas in the case of cancer cells, there are a large number of dividing cells, and they have poorly defined tumor boundaries. So these are some of the major differences between the normal cell and cancer cell. So these are some of the basic terms we have to understand before going to the topic exactly. Although the topic is on carcinogens, we are going to see that. Before that, we need to know the some basic terms in cancer. Then characteristics of cancer cells we have seen, but one of the fundamental features, the major feature of cancer is the tumor clonality, where the tumors develop from a single cell and they keep on growing. Then the increased ability to survive. This is another very important characteristic feature of cancer cell. Their increased ability to survive. If you see the normal cells, they have a stipulated lifespan. Then after that, they will be undergoing programmed cell death. That also we are going to see, which we call it as apoptosis. But here what happens is that cancer cells, they lose this ability where they will survive beyond their lifetime, increased ability to survive. This is another very important characteristic feature of cancer cell. Then aberrant regulation of cell cycle. So we all know about cell cycle. So cell cycle is very important for the regulation of cell division and multiplication and proliferation. So the eukaryotic cell cycle, we know that it has four different phases: G1 phase, S phase, G2, and M phase, isn't it? So, but in cancer, mostly the S phase is affected, the synthesis is affected, and so there is an aberrant regulation of cell cycle. Usually in normal cells, the cell cycle is regulated at the level of cell cycle is regulated by different factors. But here the cancer cells, they lose that, that regulation is very well affected in cancer cells. So the cell cycle is not regulated. So therefore, the cells get proliferated. And growth and division is, of course, the growth and division of cancer cells abnormally, there is a rapid rate. It will be in the rapid rate. And inappropriate need of hormones and growth factors. This already we have discussed. For the normal cells, we see they require the hormones and growth factors. They regulate their cell division and proliferation. But in case of cancer cells, they don't have that. There is an inappropriate need or dependency of this cancer cells for the hormones and growth factors, and they are poorly differentiated, and the communication, intercellular communication, is also disrupted in cancer cells. So these are some of the important characteristics of cancer cells. So this is just to show the difference between this is a liver cancer, which shows in order to differentiate a normal liver and liver cancer. Although physically we cannot say anything because it needs specific studies, histopathological studies and other studies to differentiate a normal cell and a cancer cell. This is just an physical outline of how a normal liver and a liver which is affected by cancer is will be there. So this is just to show the difference between a normal liver and a cancerous liver. And what are the biochemical changes? Some of the biochemical students in biochemistry, they will be able to understand this very well. Biochemical changes in cancer. So other than the physical changes, other than the morphological changes, what we have discussed about the cell size and shape, other than that, biochemically, what are the changes usually happens is there is increased synthesis of RNA and DNA in cancerous cells. So other than that, there is increased activity of the enzyme ribonucleotide reductase. I hope biochemistry students will be knowing this. Ribonucleotide reductase is a very important enzyme for the synthesis of deoxyribonucleotides. So this deoxyribonucleotides are very important for the synthesis of DNA. So there is an increased activity of this ribonucleotide reductase, and there will be also increased rate of glycolysis because because for the cancer cells, for their cell division and proliferation, they utilize more energy. So there is an increased rate of glycolysis. Because of that, what happens is that there is increased production of pyruvate. We know that the end product of aerobic glycolysis is pyruvate. So this pyruvate gets converted to lactate under anaerobic conditions, and there is excess production of lactate, and this condition is referred to as lactic acidosis. So lactic acidosis is one of the important biochemical feature in cancer. So lactic acidosis is seen. And apart from that, there is synthesis of certain fetal proteins. See, fetal proteins are usually seen at the fetal stage, like carcinoembryonic antigen. But here in cancer, what happens is that even this fetal protein synthesis increases in adults. So that is synthesis of certain fetal proteins are seen in cancer. There is enhanced synthesis of growth factors and hormones. That is inappropriate or enhanced synthesis of growth factors and hormones seen in cancer. Other than that, there is cell surface alterations because of the composition of glycoproteins and glycolipids. We know that glycoproteins and glycolipids are very important for maintaining the structural integrity of the plasma membrane. So alteration of this glycoproteins and glycolipids are also seen in cancer. So these are some of the major biochemical abnormalities that are seen in cancer. Talking about the types of cancer. So cancers are classified according to the tissue and the cell type from which they arise. It can be carcinoma. If the cancer arises from the epithelial tissues, we call it as carcinomas. Example, hepatocellular carcinoma. If it is from the mesenchymal tissues, it is sarcoma. Example, osteosarcoma. Or it is a cancer of the blood forming cells or organs, then we call it as leukemia in general. It can be either myeloid leukemia or lymphoblastic leukemia. If it is a cancer of lymph node, then we call it as lymphoma. So it is example for this is Burkitt's lymphoma or T cell lymphoma. So this is classification of the cancer based on the cell type, tissue and the cell type as carcinomas, sarcomas, leukemia and lymphoma. Next is the staging. So to see the stages of cancer, we have an early stage. Early stage is a stage 1. In the early stage, there is a small invasive mass or tumor will be seen, but there is no spread to lymph nodes or other. This is a very important point to be noted. Early stage, in the early stage, the spreading, what we call the metastasis, is usually does not occur during the first stage or the early stage. So this stage is also called as a localized cancer. That is why it's called as localized, because it hasn't started spreading. So it is also called as localized cancer. But usually the physicians say that it's very difficult to diagnose any particular cancer at this early stage, except some cancer. Generally, it is at the early stage, it's very difficult to diagnose a cancer. In the second stage, the cancer has started to affect the nearby tissue. But if we could able to detect it at the early stage, the treatment will be far better. So if you see the second stage, the cancer has started to affect the nearby tissue. Mass may have grown in size and spread to lymph nodes. So this is a very important point. It has spread to the lymph nodes. So the metastasis has started already in the second stage. Even the second stage, the oncologists say that it is okay to at least at the second stage, we need to treat the patients. If it goes beyond the second stage, where there's a regional spread, where the cancer starts affecting the surrounding tissues, that is the stage 3. Cancer affects more surrounding tissues. The mass may have grown in size. It spread to distinct lymph nodes away from the mass. So this third stage is a bit complicated stage where it affects the surrounding tissues. It has started spreading. The metastasis is very clearly seen in the third stage. So this stage is a risky. So if a patient has been identified at least at the stage 2, so the treatment modalities will be easier. So generally it is advised at least at stage 2, it is diagnosed. And the stage 4 is the distant spread. So cancer has spread to the other tissues or organs. It started affecting the other organs. For example, if the cancer is the breast cancer, it would have started affecting the lung and the liver as well in the stage 4. So beyond the region where it has originated. So sometimes this is also it's referred to as advanced or metastatic cancer. So at this stage it is very challenging, or it is very difficult to reverse. So the fourth stage is that is why it's called as the advanced stage. So usually, so this is the classification of the stages of cancer. So that the treatment will be based on the different stages. And diagnosis, yes, there are some of the diagnostic tools are CT scan, MRI scan, PET scan, biopsy, blood tests are done. Generally, blood tests are nowadays there are advanced tests like PET scan and other scans have come. So, and biopsy is also very important to identify. Biopsy is very important to identify the cell type. What types of cells are involved? Because as we have already discussed here, because the classification is based on the tissue and the cell type. So in order to classify what type of cancer, whether it is carcinoma or sarcoma, leukemia, or lymphoma, we need to do the biopsy or the histopathological analysis. Only then we will be able to identify it is what type of cancer it is. Okay. Other than that, tumor markers are also useful. For tumor markers are in general. It is not for any specific cancer. So tumor markers are there like carcinoembryonic antigen, NSC, neuron-specific enolase. These are some of the markers which are used for identifying cancer, but they are not very specific. Now, before going to the causes or about the carcinogens, there are different cases. For example, if you see the case 1, if you see the case 1, a patient, case histories, if you see a case 1 patient is a 62-year-old man diagnosed with stomach cancer. History reveals that he is a businessman. So what we understand here is, so this patient is diagnosed with stomach cancer. He is a businessman. So this is because what we understand the cause for this cancer is he's not having food at a proper time because of ulcer. They have found ulceration could have led to this stomach cancer. So this is because of the improper dietary intake. So this is because of the lifestyle or the diet. So the diet plays a very important role. So here you would have neglected food at proper time. So this has led to ulcer, and in turn it has led to stomach cancer. This may be a cause in case of this case 1. You see the case 2, a 36-year-old woman with cervical cancer. So in this case, they say that the mother already had cervical cancer. So they suspect this could be because of genetic reasons. So we say that if the first case is because of dietary reason, the second case could be because of the genetic reason, inherited reasons there may be the involvement of certain genes in the second case. If you come to the case 3, a 27-year-old youth dies of oral cancer. Now this, if you see 27 years, he is only 27 here, he is a teetotaler. So he does not have any other habits. So he's a teetotaler, but he has died because of oral cancer. So this is an unknown case. So there is no clue, because There is no genetic reasons behind this. There is no dietary reasons because he used to have proper diet. But this remains unpredictable. The cause remains unpredictable. We are not able to find out what is the real cause. But he has died in only 27 years. So these are some of the cases which explains the cause. But the third case is still we are not able to identify the reason for the third case. And now coming to the topic as such, so far we are discussing about, we are talking about what is cancer, the basic terms involved, metastasis, and the different types of cancers. Now coming exactly to the topic, carcinogens. So what are these carcinogens? Carcinogens are the agents which can induce cancer. Those agents which are responsible for inducing cancer are called as carcinogens. And the term carcinogenesis refers to the formation of cancer. So carcinogenesis is a process. Carcinogenesis is a process where how it leads to cancer. The term carcinogenesis refers to the process by which cancer is formed. So if you see this diagram, there are different stages in carcinogenesis. There is initiation stage, promotion stage, and progression stage. So there are different factors or different carcinogens involved in initiating, promoting the cancer. Can you understand that the term carcinogenesis refers to the process that leads to cancer. From the normal cell, how a cell is converted to a cancerous cell, okay, through different processes, through initiation, promotion, and progression. That is refers to as carcinogenesis. That is a general term. Carcinogenesis refers to the process, how a normal cell is turned to a cancerous cell. Okay. Carcinogens. So coming to the topic again, so carcinogens are agents, are the external agents which causes cancer. So carcinogens are divided into chemical carcinogens, physical carcinogens, and biological carcinogens. We are going to see one by one. First, we'll talk about chemical carcinogens. So how the entry of chemicals into the body. So how the body is exposed to different chemicals. Either it can be because of occupational risks. Those who are working in factories and industries, or those who are exposed to harmful chemicals like asbestos, benzene, or any other chemical. So they are on increased risk to different types of cancer. That is one way exposure, occupational risk. The other thing is dietary risk. Yes, diet like aflatoxin B1, such as intake of this aflatoxin B1, which is present in contaminated foodstuffs like contaminated peanuts. So this aflatoxin B1 can induce liver cancer. So this is present in contaminated peanuts, corn, and grains. And other important thing is the grilled meat. See, now we are talking about junk foods, and most of us, like, we consume this grilled meats, isn't it? When we go to the restaurants, we take this tandooris and this grilled meat, grilled chicken or grilled meat, isn't it? So excessive intake of this anything. So why this grilled meat can cause cancer? So what is the mechanism behind this is when we heat the meat beyond a particular temperature. What they usually do is what we call this kebab, or the grilling of meat, is done at a very high temperature, isn't it? So when there is a very high temperature, studies say that it releases, because of this high temperature, there is a release of cancer-causing chemicals in the meat. Some heterocyclic amines are released, which act as carcinogens, and so excessive intake of this grilled meats and such foods can cause cancer because of the carcinogens that are released from these foodstuffs. So that this is the dietary reason. Either it can be occupational reason, exposure, or because of this dietary reason. Next, long-term intake of drugs. Long-term, if you see, everything is long-term. Certain therapeutic drugs. For example, if you take diethylstilbestrol, this is a non-steroidal drug, estrogen agonist. So certain therapeutic drugs, when we are taking for a long time, they have the ability to get converted itself to a carcinogen. So this drugs are also very important. They play a very important role as a carcinogen if they are taken for a long term. Okay. And apart from that, we know lifestyle. So cigarette smoking is a very important cause for lung cancer, because we know that because it has benzopyrene. It's a very important carcinogen which is present in the cigarette smoke. So these are some of the reasons how chemicals enter our body, how our body is exposed to different chemicals. It can be either because of the occupational risk or diet or drugs, or because of the lifestyle. Okay, actually lifestyle can come under diet, and lifestyle can be taken together also. Okay. Now coming to the carcinogens. So we are talking about carcinogens. So carcinogens are classified into chemical carcinogens, physical and biological carcinogens. Talking about chemical carcinogens. So chemical carcinogens can be classified based on their action, like initiators and promoters. Chemical carcinogens can be either initiators or they can be promoters. And if you talk about initiators, so we have already seen, because if you see the stages of chemical carcinogenesis, it starts from initiation. So chemical carcinogens can act as initiators. So initiators can be either indirect or it can be direct. So what is the difference between an indirect chemical carcinogen and direct chemical carcinogen? The term indirect here means the carcinogen, after it has entered the body, it needs some activation. It requires metabolic activation in order to be converted to an active carcinogen. Can you understand? The difference between an indirect and direct is that indirect carcinogens, indirect carcinogens are also called as pro-carcinogens. They are not carcinogenic as such, but they get converted to active carcinogen upon activation. What is the best example for this is, if you see this compound benzopyrene. Benzopyrene is which is present in, we are talking about the smoking, which is present in tobacco smoke. Benzopyrene is present in the tobacco smoke. So this component, when it enters the body, it enters as a pro-carcinogen. But when it gets converted to active carcinogen with the help of the enzyme, we know that students from biochemistry and chemistry also may be knowing, we need this phase 2 detoxification enzyme, which is called as cytochrome P450. Cytochrome P450 monooxygenase system is very important. Actually, the role of cytochrome P450 enzymes for the detoxification, but in certain cases, what happens, they get converted. In this case, benzopyrene is being a pro-carcinogen, gets converted to its active carcinogen in the presence of cytochrome P450, which is a phase 2 biotransformation enzyme. So we can see here. So we can see here the pro-carcinogen is getting converted to an active carcinogen, the epoxide form. Then this epoxide is activated. Now this is a carcinogen, this carcinogen which can bind to DNA, and it can form DNA adducts and can cause mutation and finally cancer. So this is an example for indirect carcinogens. Benzopyrene is an indirect carcinogen which gets converted to the active form, which is the benzopyrene epoxide form, with the help of cytochrome P450. Okay, so that is indirect carcinogens. But there are also some direct carcinogens. Some of the chemicals and some of the anti-cancer drugs. See, yeah, some of the which can act as direct. They do not require any metabolic activation. So long-term exposure to such carcinogens like nitrosourea sulfonates and some of the anti-cancer drugs like cyclophosphamide, long-term treatment of these drugs may also convert the carcinogen into a direct carcinogen. So these are all examples for direct carcinogen. So under chemical carcinogen, we have initiators and promoters. Under initiators, we have indirect and direct carcinogens. So we understood the difference between the indirect and direct carcinogens. There are also promoters under chemical carcinogens. So some compounds, they promote that is which we talked about the stages, isn't it? The second stage, the next stage is the promotion. Initiation, promotion. So some of the chemicals can promote this carcinogenesis process. They are called as promoters. Some chemical carcinogens can directly promote the proliferation of the cells. Okay, they can promote they can enter at the level of this promotion stage. For example, phenobarbital, cyclamate, some of the chemicals, they act as promoters which indirectly promote carcinogenesis, which stimulate the carcinogenesis process. So this is about so under carcinogens we are discussing about chemical carcinogens. Under chemical carcinogens we have initiators and promoters. So initiators, they act at the level of initiation stage of carcinogenesis, whereas promoters, they promote carcinogenesis process. So next is so usually if you see the nature of chemical carcinogens, they are electrophiles, that is they are classified as free radicals. See, this term we commonly use, this term free radicals we commonly use under cancer because the free radicals which are deficient in electrons. So we know that free radicals are the one which have a lone pair of electrons in their outermost orbital. So these free radicals, so they play a very important role because the free radicals can covalently bind to the bases, the nitrogenous bases of DNA, and they can cause damage, which in turn leads to mutation and cancer. This is how free radical they function. So these free radicals, so they affect the nitrogenous bases and they affect DNA, they cause DNA damage, then DNA mutation, subsequently leading to cancer. So most of the chemical carcinogens are usually, they are free radicals, they are electrophiles. Okay, that is their chemical nature of this chemical carcinogens. Next is the physical carcinogens. So talking about physical carcinogens, so these are some of the examples are given in detail about this chemical carcinogens. I'm going to the next, physical carcinogens. So talking about physical carcinogens, so under physical carcinogens, physical carcinogens are the radiations when the body is getting exposed to different types of harmful radiations like UV rays, ionizing radiations, then they are classified under physical carcinogens. Okay, so they can cause cancer. Under physical carcinogens, we have we can see this is UV rays. Then the other one is ionizing radiation. So if you see UV rays, so what is the mechanism? Again, what is the biochemical mechanism behind this? If you see, again, biochemistry students must be knowing about it because we do a chapter under molecular biology when we are talking about DNA replication. We talk about DNA repair mechanisms. So here, when the body is exposed to harmful UV rays, what happens? This UV rays induce pyrimidine dimers or the apurinic sites. These are some of the biochemical terms. The pyrimidine dimers refers to formation of pyrimidine dimers, especially thymine dimers. So usually what happens, since if you see the DNA structure, according to the Chargaff's rule, adenine pairs with thymine and guanine with cytosine. That ATGC, we call it as that. We call that. But in case of this, what UV rays does is that it induces pyrimidine dimers, where adenine pairs with adenine, thymine pairs with thymine. So this is called as pyrimidine dimer. Sorry, not adenine. It is a thymine. Thymine dimers are formed, or sometimes cytosine dimers are also formed. But most common dimer formation is thymine dimer in the same strand of the DNA. This is also called as apurinic site. The term apurinic refers to, instead of a purine, again a pyrimidine is added along with a pyrimidine. So it's called as, that's why they are called as pyrimidine dimer or apurinic site is formed because of UV rays. So UV rays induce apurinic site or a pyrimidine dimer in the DNA. This causes mutation in DNA and it leads to cancer. So this is the mechanism how a physical carcinogen like UV rays can cause cancer. We see the other side, ionizing radiations. If the body is exposed to ionizing radiations like gamma rays and X-rays, there is enhanced, again, free radical production. These are radiation, they increase the free radical production in the body, which reacts with DNA. This mechanism we had seen. So these free radicals, they affect the nitrogenous bases, which causes DNA damage, mutation, and cancer. So this is how the physical carcinogens, they lead to cancer. So we have understood the mechanism of UV rays and the ionizing radiation. So this is this representation shows the difference, the mechanism behind the action of physical carcinogens. Okay, so example for this is, the best example for this is, so there is for this pyrimidine dimer, we were talking about pyrimidine dimers. Yes. So yes, there see the body has a mechanism to rectify it, which we call it as DNA repair mechanism. If a pyrimidine dimer is formed, there are different DNA repair mechanisms to rectify these defects. But if there is a genetic defect in rectifying that, if you see this condition, which is called as xeroderma pigmentosum, so this is a skin cancer, which is because of a defect in DNA repair mechanism, okay, which is called as nucleotide excision repair mechanism. This is because of the presence of a pyrimidine dimer, which we have discussed here. Pyrimidine dimer caused because of UV rays. Although here it is it's a genetic disorder because of the enzyme defect. So such patient should not be exposed to sunlight because photosensitivity is a very important characteristic of xeroderma pigmentosum. So this is a example for a skin cancer because of the defect in the DNA repair mechanism, which is not able to correct the pyrimidine dimers. So physical carcinogens can induce. Why this is shown is they can induce this, have the ability to induce skin cancer, xeroderma pigmentosum. Okay, so this is about the physical carcinogens. Next is the biological carcinogens. So under carcinogens, we are talking about, we are discussing about different types of carcinogens. Carcinogens are the agents which induce cancer. Carcinogens can be either chemical carcinogens or it can be physical carcinogens. We have seen the example for chemical carcinogens. There are different chemical carcinogens. It can be either physical carcinogens like UV rays or X-rays and other harmful radiations. There are also biological or biologic carcinogens. So under biological carcinogens, we have bacteria and viruses. Yes, viruses, they play a very important role. They are the most significant role players in causing cancer, viruses. If you ask bacteria and yes, bacteria can bacteria cause cancer? Not directly, but maybe indirectly. If you take, must be knowing, even microbiologists may be knowing that there is a Helicobacter pylori infection. This Helicobacter pylori is a bacteria which causes the infection of the stomach. If this is not treated, it leads to ulcer, and this ulcer can in turn cause the stomach cancer or the colon cancer. So this is an example where a bacteria can cause cancer. Example is the H. pylori infection. If this infection is not treated, it can even develop into a cancer. So that is an example, but mostly viruses have a major role to cause cancer. And these viruses, we call them as oncogenic viruses. They contain either DNA or RNA as their genome. See, why we talk about viruses is even in, if you see this coronavirus, so it is coronavirus is also an RNA virus, isn't it? So similarly, these viruses are very harmful. So they can even cause cancer. So these viruses can be either DNA virus or RNA viruses. So for example, DNA viruses like papovirus, herpes virus, and pox virus, they can cause cancer. The RNA virus, they are retroviruses. We all know that they have this reverse transcriptase enzyme, and they use the host RNA for producing the viral DNA. Okay, so example for RNA virus include the sarcoma virus, leukemia sarcoma virus, and other mammary tumor viruses. And the mechanism which is understood under virus-induced cancer is maybe because of by the activation of oncogenes. We are going to see just what are the genes involved in leading to cancer, that we will see briefly. By activation of either oncogenes or by inactivation of tumor suppressor genes, or affecting signaling pathways that leads to cancer, or chronic activation of some of the inflammatory responses. So these are some of the possible mechanisms behind virus-induced cancer. Okay, so this is about the, so this table explains the different types of DNA and RNA viruses and what are the cancer it can produce. Okay, so these viruses are in turn, in general, they are called as oncoviruses, which have the ability to cause cancer. It's not that all viruses can cause cancer. Those viruses which have the ability to cause cancer, they are classified as oncoviruses. Okay, so this is about biological cancer. carcinogens. So we are under carcinogens, we have discussed about chemical carcinogens, physical carcinogens, and biological carcinogens. Okay. So it was a myth that it's the disease of the poor. It was in the ancient days, it was understood that cancer patients with cancer are poor people. It is usually only poor will suffer from cancer, but it's not like that, because we know many celebrities like Manisha Koirala, we know this batsman Yuvraj Singh, they all had cancer, and these people have recovered. We know that this actress, she had this ovarian cancer, and Yuvraj Singh, he had lung cancer. So because of different causes, and so they were treated. Actually, they had undergone treatment, and they have recovered from this disease. So this is not the poor. Maybe it's actually a myth. It's not a disease of the poor. Maybe because of cancer, the person may become poor, because see, it is said that the treatment facilities, see, the treatment for cancer is very expensive. So the cancer can make a person poor, but it is not a disease of the poor. This is to be understood. Okay. Then the molecular basis of cancer, just about some of the genes which are involved in cancer, because some of the cancer, they say about 10 to 15% cancers are hereditary. We call this the inherited cancers, or because of the hereditary reasons. So the molecular basis of cancer, some of the genes related to cancer, proto-oncogenes, and tumor suppressor genes. So if you see proto-oncogenes as such, they are not cancerous, but proto-oncogenes may be converted to oncogenes. So the difference between proto-oncogenes and oncogenes is that this proto-oncogenes are the normal genes which get converted to oncogenes, like this RAS, c-MYC. I'm not going deep into the topic as such, because just to know that there are certain genes which are involved. So oncogenes are the genes which get mutated. Proto-oncogenes are the normal genes. They get mutated to oncogenes. The other important one is this tumor suppressor genes. So in the body, we have a set of genes which are called as tumor suppressor genes, which have the ability to suppress tumor, because these genes will produce proteins which act as factors that can suppress a tumor. For example, P53 is a very important tumor suppressor gene, which is called as guardian of genome. If this gene is mutated, then inactivation of this tumor suppressor gene is one of the important causes for cancer. Okay, tumor suppressor genes. And other important thing is the genes that regulate apoptosis. So apoptosis is also called as a programmed cell death. We all know that the normal cells, they will undergo apoptosis after a certain period, but the major difference between the normal cell and the cancerous cell is irregular apoptosis mechanism in case of, or the disruption of apoptosis mechanism in case of the cancerous cells. So apoptosis mechanism is a very important factor to regulate this carcinogenesis process. In case of normal cells, that is obeyed, but cancerous cells, they do not obey this apoptosis mechanism. So they deviate from this. And there are some of the proteins like BCL2, BAX, caspases, which are involved in the regulation of apoptosis, that signaling pathways. So in cancerous condition, all these pathways are dysregulated or disrupted. Okay, so this is a brief idea about the role of the genes related to cancer. Okay, so before we complete, we just experimentally see, there are now research has been extensively carried out in vivo models as well as in the in vitro models. So if you talk about the in vivo means within the body. That is, in vivo models, different animals, experimental animals are used, like mice, oyster rats, and guinea pigs are used for the in vivo models. And in vivo models are very useful for the drug design. See, why in vivo models are carried out is to understand the molecular mechanism in the carcinogenesis process. The importance of this in vivo model, using in vivo models for experimental cancer, is to understand the molecular mechanisms as well as for the treatment. So this will also help us for the treatment. It will give us an idea of the treatment strategies for cancer. So these are the animals. So some of the drugs, carcinogens, since we are talking about carcinogens, there are some carcinogens which are used for inducing experimental cancer. In the laboratory, these carcinogens are commonly used for inducing experimental cancer. For example, benzopyrene is commonly used for lung cancer, for inducing lung cancer in animals. Then diethyl nitrosamine is used for liver cancer. DMBA is used for breast cancer, and this 4-NQO NQO is used for the oral, for inducing oral cancer. So this, so why I am talking about this is because the recent days, so research has been carried out with the help of these carcinogens. So these carcinogens are proved to be extremely carcinogenic, and they induce different types of cancer. Okay, this show how this cancer is induced. So usually carcinogens, they are given either as oral route, depending on the solubility. It can be by the orally, it is given to the animals, or it can be intraperitoneal or intravenous. So these are different modes of administration of drugs. So before concluding today's topic, so the dietary importance. So cancer and diet. So how diet plays a major role. This we had already seen in the previous slide, that how diet is very important for the prevention as well as management of cancer. So diets, when we talk about free radicals, because we have seen that free radicals are very important in inducing different types of cancer. So when we increase the diet rich in antioxidants, now this term is very commonly used even by a layman. Even common people, they are using this term antioxidant. And if you purchase any items, whether it is an oil or a cream, it's a face cream or it's a body lotion, wherever you can see that the term antioxidant is used. So these antioxidants, so generally what research says is that increased intake of antioxidants prevent not only cancer, any type of disease. To protect ourselves from any type of disorders, diseases. So this antioxidants, they play a vital role in that. So increased intake of antioxidants like vitamin A, E, C, curcumin, which is present in turmeric, and increased fiber-rich diet. So all these things, they play a major role in preventing cancer. And obesity and cancer, yes, of course. So they say that about 7 to 8% of cancer. Is there a relation between obesity and cancer? Yes, of course. About 5 to 8% of cancer are caused because of obesity. Although the direct mechanism is not understood, it could be because of either increased insulin-like growth factor, increased secretion of insulin-like growth factors, decreased DNA repair capacity. In case of, I'm talking about obesity, how obesity is related to cancer. These are some of the mechanisms which are understood, either because of this insulin-like growth, IGF, increased IGF, or decreased DNA repair capacity, or increased DNA damage, which is found in obesity. Another important thing, what the research is saying is adipose tissue. Adipose tissue also creates an inflammatory environment that enhances the ability of these tumor cells to metastasize. So adipose tissue creates an environment for the tumor cells to metastasize. It enhances metastasis. So that is how obesity is related to cancer. So lifestyle is very, very important in the prevention of cancer. So these are some of the foods that are given fighting against cancer. So diet plays a major role in combating cancer. So it is advisable. So always prevention is better than cure, as a proverb goes. So with this, I want to end the topic by saying this: prevention is better than cure. So when we increase our diet with rich in antioxidants, so it has the ability to fight against these free radicals. And diet rich in antioxidants have proven to be not against only cancer but so many other diseases. So we have to have the diet foodstuff that are rich in antioxidants. So what's new in this? So recent researchers had given a clue on how diet rich in fiber reduces. So one article says that, I have not quoted the article here, the journal. So one study says that diet rich in fiber reduces the risk of breast cancer. Some studies say that fasting along with vitamin C treatment is very effective for for treating some other type of cancer. See, these are supplements. Not all the direct effects they have, they are all used as a supplementary treatment methods. And adding more spice to our diet may also reduce the risk of cancer. These are some of the ongoing research on cancer. So with this, I would like to end my session. So I once again thank the management of Anna Violet Arts and Science College for giving me this opportunity to interact with the students, faculties, and other various scholars. Once again, I congratulate and appreciate the organizing team behind this. So I thank the principal, Dr. Kamala Balakrishnan ma'am, the convener, Dr. Parimla, because she was there behind me like for the last 2-3 weeks about organizing this. So I really want to thank her and the co-conveners, Dr. Prabhakaran, Dr. Chandramohan, and the whole entire team and the departments which are organizing this, the chemistry, physics, and the biochemistry department. I would like to congratulate, and I would like to convey my best wishes for all these departments. And I hope that we'll have many more interactions and sessions like this in the future. Thank you all once again.
Thank you, sir. Thank you very much for your valuable information about carcinogens. Now it is the time to discuss the questions posted by our participants. Shall we discuss, sir?
Yes, please. Yes, please.
Okay. Thank you, sir. The first question is cancer a genotype or a phenotype?
Okay. Genotype or phenotype. Okay. See, actually, we have discussed about both the possibilities, right? We have about 10 to 15% of the cancers can be of the genetic nature. Other than that, we have seen about the exposure to the carcinogens. So it both the ways it is possible, because there are also hereditary cancers, are also inherited cancers. As we have discussed, even a newborn has a cancer because of the inherited reasons. So it can be of both the natures. It can be both genotype or as well as phenotype. Both are possible. Phenotype, when we are exposed to different types of carcinogens, whether it can be chemical carcinogens or physical or biological carcinogens, then the risk is higher. So in both the ways, it is possible.
Okay. Then the second question is thioacetamide is a carcinogen.
Okay.
Whether it is a tumor initiator or tumor promoter.
Thioacetamide.
Acetamide.
Okay. Thioacetamide. Usually, thioacetamide, I think it should be an initiator, because promoters are usually less. Promoters, some of the promoters like phenobarbital and cyclamates are being known carcinogens. So thioacetamide, although I'm not sure it should be an initiator of carcinogenesis.
Okay, sir. Sir, whether all epoxides are carcinogens, sir?
Yeah, epoxides. Yeah, this is also very good question actually. So usually when a procarcinogen gets converted after metabolic activation, it becomes an epoxide. So these epoxides are generally thought to interact with DNA. Mostly these epoxides are DNA, and they damage DNA. They cause DNA damage and mutation. So mostly epoxides are yes, they are carcinogenic, and they are actually these epoxides are the major, we can say they're the key role players in causing cancer. So they have a major effect on DNA. Yes, so epoxides, so most of the epoxides are carcinogenic. Yes, they can cause cancer. They can lead to cancer.
Okay. How to determine if a genetic mutation is a driver mutation for a specific tumor?
How do we determine genetic mutation?
Is a driver mutation for a specific tumor.
Okay. How do we determine? Okay. Okay. This is also a very important question. Actually, if you see some cancers like breast cancers, in this breast cancer, we have the breast cancer, and actually there are genetic reasons. For example, this BRCA1, it's a very important gene which is involved in inducing breast cancer, and extensive research has been taken place on the study of this genetic mutations, because because we are not exactly, the cause is not known. So this genetic studies are usually recommended for understanding whether the cancer is inherited reasons or it is because of the because of the exposure to carcinogens. So it's especially, as I told you, in case of breast cancer, it is the BRCA1 is usually identified, but still research is on this. So extensive research is on to understand the molecular basis. So because genetic mutations, we have to understand at the molecular level. So extensive research should be done to understand the type of mutation, especially the genetic mutation.
Okay, sir. Thank you, sir. Thank you very much, sir.
Okay, thank you.
We have come to the end of this section. I thank everyone who joined us for this today's session. We are extremely thankful to our Dr. Anandakumar Pandey, Assistant Professor, for his gracious presence in spite of his extremely busy schedule, which is a reflection of the importance that is attached to the commitment of his work. We are all inspired by your informative lecture on carcinogens, sir. Thank you once again. I also extended our thanks to our delegate guests. Now I request the participants to fill the feedback forms given in the chat box and posted it to get the e-certificates. Participants, please stay back with us and join the next session by